Hello, and welcome to today's webinar, Case Studies in Foreign Particulate Analysis, presented by Gateway Analytical. My name is Brittany Norris, Marketing and Creative Director for Gateway Analytical, and I'll be moderating today's event. Before we start, I'd like to go over how you can interact with us today. All lines will be muted during the presentation to ensure good sound quality and minimal background noise. You can submit a question at any time using the chat functionality in the lower left portion of your screen. You can also interact with us on Twitter today at #GatewayWebinars. At the end of the webinar, you'll be asked to take a short survey. After the webinar, you will receive an email that includes a link to the webinar replay, the presentation slides, and a digital e-certificate of attendance. We've also allotted time today at the end of the webinar for Q&A. Before I introduce today's presenters, I would like to provide everyone a quick overview on Gateway Analytical. Gateway Analytical is a full-service analytical testing laboratory that features specialized expertise in particulate identification. We provide detailed analysis to characterize and identify particulate, in addition to providing extended customer support to help identify the source of contaminants. At Gateway, we know the challenges associated with identifying particulate, and our focus with each customer is to lead them to fast remediation, because we know particulate happens, but that shouldn't stop your progress. Now let me introduce you to today's presenters. David Exline, Vice President of Gateway Analytical, and Julie Patterson and Lorena Parra, both scientists at Gateway Analytical. David has more than 20 years of experience in managing and administering analytical laboratory and consulting services. David has specific expertise in applying chemical imaging methods such as Raman, UV-Vis, and near-IR techniques to solve the most difficult pharmaceutical and material science problems for Gateway Analytical customers. David is adept in numerous pharmaceutical-related services and testing methods, including materials characterization, particulate contamination analysis, particle sizing, and high-resolution microscopy. Julia started her career in 2010 as a scientist in the biotechnology and pharmaceutical services sectors. She has over 5 years' experience in performing materials characterization and in the identification of foreign particulate matter. She has taught several classes to both pharmaceutical companies and the FDA on the management of foreign particulate matter in pharmaceutical processes. Lorena Parra has more than 3 years of experience in materials characterization analysis. Lorena has worked as an SEM technician for a year before joining Gateway Analytical, and also provided mineral characterization services during her time as an undergraduate. As a scientist, Lorena focuses on providing particulate analysis and materials characterization to Gateway Analytical's customers. Welcome, everyone. Hi. Hello.
Hello.
Now I'm going to go over a brief outline of today's webinar. First, we'll begin with a brief definition of foreign particulate matter, along with some common sources, a comparison of manual and automated analysis, an overview of sample preparation and analysis methods, including specialized analysis methods, and then finally our case studies. So first, what is foreign particulate matter? Foreign particulate matter can be visible and subvisible particles. Often, especially on tablets, they will appear as stains and discolorations. They can be charred from having gone through an autoclave or similar processes. They could be precipitated, drug product, or other materials. And they can be found at any stage in the manufacturing process. So why is foreign particulate matter so common? Well, the manufacturing process has many stages, and your product is in contact with that process at every stage. But also during R&D, you can see chemical breakdown and aging producing particulate matter. There can be precipitation and contamination at a variety of stages in the process. It's important to remember that sterile does not mean particle-free. So even working in a sterile environment does not prevent particulate contamination. So this is not an exhaustive list of types of foreign particulate contamination in pharmaceutical manufacturing, but these are some things we very commonly see, especially stainless steel polymers and natural fibers, which again can be found at any stage in the manufacturing process. So now we have a quick poll question. When you need to identify foreign particulate, do you usually handle the analysis in-house or do you outsource it? Looks like a lot of you, there's a good mix here. A lot of people handle it in-house, but there's a good mix of people doing both as well. All right, so in order to solve particulate analysis, we like to use a process called pharmaceutical forensics, which is the application of forensic test methods and problem solving focusing on root cause analysis and identification of unknown materials as it relates to pharmaceutical products, processes, and devices, with the aim of answering questions like, what is this foreign material, where did it come from, and this can be helpful in all kinds of nonconformance investigations and similar problems. We have another poll question. What types of instrumentation are you familiar with using for foreign particulate analysis? Automated Raman, Fourier transform infrared spectroscopy, HPLC, PLM, Raman spectroscopy, and SEM-EDS. Oh, wow, okay. Looks like a lot of you are familiar with FTIR. That's good. We don't have to spend a lot of time on that. And just about the same number of you are familiar with SEM. That's good. We don't have to spend a lot of time on those methods. Now I'm going to pass it to Julia for a quick overview of the differences between manual and automated analysis.
Okay, so as Marina explained, we follow a forensic approach to sample analysis. And this flowchart depicts the route of analysis that may occur with a sample based on the sample itself and what you are looking to get out of the analysis. So up in the top part of the flowchart, there's the blue boxes. Those consist of some of the manual analysis methods that we perform and that we'll talk about today, including glass delamination. And in the green boxes on the bottom, those are some of the automated analysis methods that we'll go over, including CCSEM and the SPE instrument, which performs automated Raman analysis. So the choice of using a manual versus an automated analysis method is going to be determined by the sample itself and, again, what information you want to obtain from that analysis. With manual analysis, you're going to be able to determine the size, shape, and color of each individual particle that you're analyzing, along with both the elemental composition and spectral data for that sample. Therefore, manual analysis is good for determining the identification of particulate. However, manual analysis is generally more time-consuming, so it's going to be better suited for a smaller number of samples or for a situation when you're interested in analyzing only a representative sample from a larger population. With automated analysis, you're going to be able to analyze a larger amount of particles in a shorter amount of time. However, sometimes you will not be able to obtain as much data from each individual particle, depending on what analysis method that you choose. So automated analysis is usually more suitable when you need information or characteristics of a population of particles, or if you're looking at trending data. So first, I'm going to discuss the techniques involved with some of our manual analysis methods. And this is a flow chart of a breakdown of basically the route a sample would take for our standard particulate analysis. So first, a nonconformance issue will occur. And after that's identified at the site, the sample can be collected and packaged in order to be sent for analysis. After the sample is received by the laboratory or department performing that analysis, the sample is further isolated and prepared. During that preparation, the sample's physical properties are going to be observed, like the color, shape, and texture of the particulate. and then further microscopic and optical evaluation is going to be performed, followed by the rest of these manual analysis methods. And the analysis methods that are performed are still going to depend on the type of sample that you have, because some samples lend themselves better to certain analysis methods. And then all of the analysis data is going to be compiled to identify the foreign particulate matter. And if a reference sample is sent, that reference sample can then be compared to the unknown to perform a source determination. So if we follow that flow chart, the first step is going to be the initial collection of the sample. And the isolation method will depend on where the particulate was identified. So if a particulate is located in a liquid or in a liquid drug product, then you're going to filter that drug product usually in order to isolate the particulate out. Manual isolation can also be performed, and that would include things like using a pair of forceps just to pick off the particulate or fibers from the substrate, or things like residues could be collected using a plastic blade or a swab. And then you're going to want to package that particulate in a way so that it's not going to be lost or contaminated during transport. After initial isolation, the particle is going to then be prepared for further analysis. And it's important to note that if a particle was located or had contact with a drug product, we will generally rinse that particle with DIUF water, and that's deionized ultrafiltered water. And this minimizes interference from any residual drug product that could be on the surface of that particulate after isolation. This slide just shows a few pictures of a typical filtration setup. And the filter material and size is going to be selected based on the sample and the liquid. Typically a polycarbonate filter is used, and the size of the pores is selected to be smaller than the size of the particulate that you wish to capture. Some other filters, like gold-coated filters and nylon filters, can also be used under special circumstances. And the sample liquid can be pipetted directly onto the filter, as is shown in these pictures, or you can pour the liquid into a funnel that will concentrate it on the filter. For manual isolation and transfer, everything is going to be performed under a hood. And this is especially important because we are usually dealing with very small particulate. So you want to have that particle-free environment. And in these images, you can see a scientist transferring a particle from a filter to an SEM stub using a stainless steel probe. Other stainless steel tools like forceps and scalpel blades can also be used. And it just takes practice to handle these small particulates, and it takes practice to understand which tools are best for the different scenarios you have. So when we do manually prepare a sample, it is then going to be transferred to the appropriate substrates for the analysis being performed. And here we just have a few pictures of the substrates that we most typically use for our manual analysis methods. For microscopy, temporary mounts are usually made with glass slides and glass cover slips. And the temporary mounts are used because they can then be taken apart to prepare the sample for further analysis. For FTIR and Raman, we use slides that are coated. So for FTIR, we use a low E slide, and that coating is not FTIR active, so that gives us very little background interference when we're collecting our sample spectrum. And samples are actually flattened onto that slide using a stainless steel roller tool. Raman samples can then be placed directly onto an aluminum slide, so they don't need to be rolled out or anything like that, and the aluminum is not Raman active.
For SEM analysis, samples are just placed directly onto the SEM stub, and that stub has a double-faced carbon adhesive on the surface. That's that black surface that you see in those pictures. And a piece of filter can also be transferred to SEM stubs for manual or CCSEM analysis. For automated Raman analysis, using our SPE instrument, samples are simply filtered onto a gold-coated polycarbonate filter. And this gold coating is not Raman active. So again, that's just to reduce any background interference. And those filters can go right into the instrument. So, are you currently in the process of trying to identify foreign particulate? And since many people do perform it in-house, it looks like there is a good number of people that are in the process of trying to identify something. It looks like there's a good amount too that may be between investigations. So the first manual analysis technique that I'm going to discuss is microscopy. And we utilize many microscopes here at Gateway Analytical. We have stereo microscopes, polarized light microscopes, fluorescence microscopes, and comparison microscopes that we can use. And as you can see listed on this slide, there are many features and properties of a sample that can be observed by using microscopy. And the characteristics of a material, either under optical or polarized light microscopy or fluorescence, can help us to characterize particulate. And you can get a lot of information about the sample just by looking at it under the microscope, especially with samples like fibers. However, these techniques become even more important when you're comparing an unknown to a reference material. So for example, you might have two cotton fibers that look similar, but when you put them under the fluorescence microscope, one may fluoresce and the other may not. For our FTIR, or Fourier transform infrared spectrometry, we typically use the microscope attachment for our FTIR analysis. Although other methods like ATR or the salt plates could be used, we use the microscope attachment because we're dealing with such small sample sizes. And I won't go too in-depth into FTIR since it looks like a lot of you are familiar with it, but light is going to interact with the sample, and that interaction is interpreted as an FTIR spectrum. And FTIR analysis is good at identifying things like polymers and fibers, among many other samples. However, there's a few limitations to using the FTIR instrument. For example, the FTIR can't resolve mixtures. So if you do have a mixture of substances, the spectra for those two substances are going to show up in one spectrum. So it's up to the analyst to kind of tease out that spectrum to try to find the different components of a mixture, and that can get pretty difficult. Also, some compounds like straight-chain hydrocarbons, fats, and oils, it's very hard to get a more specific call on those. It can be done, but it depends on the sample. For Raman spectroscopy, it's generally used as a surface analysis method, and you're able to see the sample in the instrument and direct the instrument's laser to a specific area of the sample. The laser will then interact with that area of the sample's surface and yield a spectrum. So this makes Raman analysis good for embedded materials that cannot be isolated. So for example, if you have a material embedded in a tablet, you could obtain a spectrum just from that material in the tablet still, as long as that material is exposed. It's also good at picking up pigments and oxides and for identifying amorphous carbon, which can be indicative of a thermally degraded material and can be encountered frequently in forensic processes since some components are going through some very high heat areas. And Raman spectroscopy can be a complementary technique to FTIR analysis. So for example, you may have a blue fiber that shows up as polyester with an FTIR analysis, but Raman analysis may yield a spectrum for a blue pigment that is part of the fiber dye. So it can definitely help you to further analyze the sample with FTIR. And SEM-EDS analysis with the scanning electron microscope is another technique that we saw a lot of you have experience with. So for manual SEM analysis, the analyst is going to manually drive the instrument and collect data from a sample. The manual analysis is good for determining surface features and elemental composition of a sample. It's also very good at imaging and analyzing different areas or regions that may be present in a heterogeneous sample. And in the images on the left, you can see the shape and surface features of the sample. And on the bottom, you can see the elemental spectrum that was collected from the sample. And actually for this sample, if you look in the image where my green arrow is pointing, we were actually also able to identify that those black spots were carbon rich. So there were two different areas on the sample. For automated CCSEM analysis, which is computer-controlled SEM analysis, the analyst is going to tell the instrument what area to analyze, and the analyst will also determine the analysis criteria for an automated run. So you can set up a run to analyze only particles in a certain size range or to classify particles in size bins. You can also count only particles of certain elemental compositions, or you can set it up to do both. This makes automated analysis good for collecting elemental data and collecting size data for a large population of particles. And in the picture on the left, you can see a run in progress. The instrument is scanning the sample area in different quadrants that it made, as you can see on the left with the green grid. The yellow highlighted box that's here is actually the area that's currently being analyzed, and that this box on the right is the area that the instrument's currently scanning. The green particles in that box are particles that the instrument flagged for analysis. And below that, you can see the spectrum of the current sample being analyzed and an image of that particle. And I'm now going to turn the presentation over to Dave to review some of our specialized techniques of glass delamination and automated Raman analysis.
Thank you, Julia. Okay, what we're going to talk about just for a minute here is some of the specialized. Julia talked about manual and automated scanning electron microscopy. We certainly can apply that to some of these newer specialized areas that we're encountering a lot in the pharmaceutical industry. A large problem right now, especially within the parenteral drug marketplace, is glass delamination. For the last several years, we've been interacting with a lot of our customers as well as the glass manufacturers to try to help address this for a lot of customers. So I just wanted to give a brief overview of what it is and kind of how we take a look at this and how it relates to the USP 1660 specification as well for that compared to kind of routine glass delamination analysis. So what is glass delamination? It's basically the formation of glass lamellae on the interior portion of the glass containers. And these eventually can be shed and delaminate off of the interior surface. And this allows small glass particles to be let loose and into the drug product. So there's definitely a few different methodologies that we're doing to try to address these issues. How does it occur? It's basically an effect of glass corrosion. What's happening basically as you have a drug product that may be slightly acidic or slightly basic or even in a neutral environment, interacts with the inner surface of the glass material. And there's some dissolution of some of the elements that might be in the glass, which leaves a silicon dioxide kind of rich region on the interior surface of the glass. And this becomes a weakened area and starts to allow these two glass particles to flake into the product. There's multiple methods and strategies for addressing these. There is a USP methodology, 1660, and I just wanted to give a real quick distinction. USP 1660 is basically for a pharmaceutical manufacturer that is testing the stability of their drug product in a particular type of vial. So it's a very good methodology if you have a drug product and you want to understand what a suitable vial will be. USP 1660 gives a nice stability type of a test for you to be able to go through and evaluate those. And then there's different analytical methods that if you have a baseline and you want to understand, you can apply things like ICP mass spec or conductivity, things like that to understand how over time your product is reacting. What we often deal with as well is just a drug product that has glass lamelli in them, has a delamination issue, and the question is to just identify whether glass delamination is happening. In that case, typically what we will do will be kind of modify the USP 1660 and select some of those methodologies, which would be stereo microscopy, optical microscopy, and scanning electron microscopy of the glass lamelli or the interior surface of the glass surface. Another specialized methodology that we utilize here at Gateway, which is kind of a newer technology, is an automated Raman and LIBS technology. And as Julia talked about earlier, there's different analytical techniques you use to solve different analytical problems. Some require a manual when you need a very deep understanding of a single particulate, and then there are some automated methodologies that you can apply when you need to understand the general overall population of the particles in a sample and get some general understanding of their characterization and not have quite the thorough understanding of doing particle by particle. What this allows us to do is look at hundreds or thousands of particles, have a very distinct particle size distribution, and also be able to give a general idea of the type of particles that we're dealing with. We utilize automated Raman for organic type of materials, foreign particulate or API or excipients, and then we can utilize automated LIBS or the automated CCSEM or computer-controlled scanning electron microscopy that Julia discussed to talk about the inorganic portion of these materials. And basically, the next slide, and you'll get a copy of these slides, they'll be accessible to you, and you can go through this in more detail just for the saving of time. The idea of this is just kind of a matrix. So at the end of the day, automated methodologies are very good for sizing and counting and giving you chemical information at a very high level. If the question is specificity of the type of material, then a lot of the manual Raman or manual optical microscopy or IR are much more effective methods. The real benefit is speed and cost when you look at manual versus automated, especially as we deal with automated Raman spectroscopy. A single analysis can cost $500 to $1,000, for example, where if you do an automated analysis, you may be able to get an idea of your population of particles for a little bit more than that, but not 100 times that amount. So it's certainly something that if you're looking at larger populations of particles, it's an effective methodology to do that. Here's kind of an idea of what that looks like from an automated. Julia had shown you a picture a little bit earlier of a filtration system on a gold-coated polycarbonate filter. So typically, a sample is rinsed onto one of those gold-coated polycarbonate filters, and as you can see on this left image here, we can see what it actually looks like as a montage of the optical picture, and then each of those particles is made into a binary image, which allows you to do particle sizing to that sample. Once we've determined our particle size, we can go back to each of those individual particles and do Raman spectroscopy. And so now we understand the size of the particle, and we now also can look at the spectral profile that will happen. That is then corrected and searched against an automated database and gives us a lot of statistical information about a sample. So that information can be binned from different size ranges. It can give us an overall particle number, a particle size distribution of the particulate that are there, and an identification of the particles that were selected for identification. So this is a great method to use for understanding particulate contamination in an organic perspective, and then utilizing automated LIBS or automated scanning electron microscopy. Very effective for using it for the inorganic portion. So I just wanted to go over a few of the specialized types of particle characterization techniques that are utilized. And now I'm going to pass it to Marina to start talking a little bit about some of the case studies. Thank you.
Okay, so I'm going to start with a very common problem that a lot of people have with an internal case studies on unknown clear particles. So clear particles are very common, and they can be really a wide variety of things. They could be glass, it could be tiny pieces of polymer, cellulose, drug products. Sometimes you get salt in there. You just don't know. Optical analysis can give you some idea, like you might be able to see fracture marks on glass, or you might see rounded edges on polymers, but often further analysis is required to differentiate between the different types, including generally SEM, PLM, FTIR, or Raman, depending on the sample specifically. So we have a quick poll question. Have you ever encountered an issue with identifying clear particulates? And it looks like a lot of you have, at least to some extent. That's good. We'll be talking a little bit more about that. So here's an example, a very common example, an unknown particle on a filter membrane. You can see it's clear. It's this little guy right here, in case that's not clear. It has kind of rounded edges. What could it be? Well, by trying to manipulate the particle, you can tell that the texture is not hard, so it's probably not glass. And we were able to cut it to do SEM and FTIR. SEM told us that it was carbon rich, which means that it's not glass for sure, because that would be silicon rich, and it's not salt, because that would have different elements. And then doing FTIR told us that it was polyethylene. Depending on what your particle feels like when you manipulate it, that'll tell you what type of analysis you should probably at least try, and then proceed from there. Now I'm going to move on to an example that was a little more challenging, an embedded particle in a vial. No, going back. Okay, so a customer had an issue. Vials were flagged during visual inspection because they had a black and brown material on the side of their vial walls. And because of where it was located on the vial, they couldn't extract it. So they were sent to us. We broke the vial in order to expose the unknown material and determined that it actually couldn't be extracted because it was embedded. Because it was embedded, surface techniques like SEM and Raman were really all we could do, but thankfully that was enough. Here's a quick picture of what the embedded sample looks like. And then here's SEM over here. This is the material. And SEM told us that it was iron and oxygen rich, and then Raman said that it was an iron oxide. Iron oxide particulate is common in glass vial mold materials, which at least gave this customer somewhere to start. So now I'm going to pass it back to Julia for one more case study.
Okay, so this case study involves the manual analysis method of microscopy. but we are going to go one step further in performing an analysis of an unknown hair. So hair can be persistent in some processes because it has a high resistance to degradation. When an unknown hair is received, we can tell a variety of information from it in order to assist us in its identification. And some of these properties that we can look at to help us identify the hair are listed on the slide. I won't list them all out. And it's important to note that we can usually speciate, or at least tell if the hair is animal or human in nature, but a forensic hair comparison to a reference sample is required in order to associate that hair to a specific source. So, has anyone ever encountered an issue with finding hair in their process? So it looks like, well, they're kind of evening out. It looks like there's a good amount that have. There's many of you that aren't sure or that haven't encountered it, but because of the clean room policies that most places have, hopefully it is a rare occurrence. So in this case study, a customer found a hair in their manufacturing process. They were able to isolate it with forceps and package it between 2 glass slides in order to send it out for analysis. So as you can see, it's a pretty big hair. And we were able to utilize polarized light microscopy to confirm that the sample was in fact a hair and that it wasn't a fiber or some other type of material. And we were also able to determine the species of the hair. This is important to determine because whether or not it's human or animal and what type of animal it could be could affect any corrective actions taken. So using our PLM, we were able to get these images of the hair. So you can see in the first box, it had a spade-shaped root, and it also had a wide amorphous medulla, which is shown in the second box. In the third box, you can see that there was some evidence of physical damage in the form of splitting, and that's that white line that you see down the middle of the hair. It was actually starting to split in some areas. And then in the fourth box, you can see the narrow, tapered distal end, and that's basically the end of the hair farthest away from your head. And we were able to identify, based on all of these properties, that the hair came from a canine. A definitive source as to the exact animal that it came from could not be determined due to no reference being submitted for comparison. However, since we were able to speciate the hair as a dog hair, the client was able to determine that it probably would have come from a piece of clothing or an object brought into the building, since there's a very, very low chance that a dog was running around in the laboratory. So I'm going to turn the presentation now over to Brittany to start some of our Q&A.
Thank you, Julia. So thanks, everyone, and that concludes our presentation segment of today's webinar. And now I'll be opening up the session for the Q&A. Remember that you can submit your questions at any time. We already have several questions in the queue, so I'm going to go ahead and get started. So first question is sort of a series of questions, and I'm going to just put them all together. So how do you isolate the particulate, how do you isolate the particles without being contaminated? And do you have to have a clean room facility to study foreign particles under a microscope? Additionally, what spectral libraries, Raman and IR, do you use for particle ID? Who would like to take the question?
So I'm going to address the isolating the particle without contamination. So basically, from a particle isolation standpoint, all samples are analyzed in a laminar flow, particle-controlled environment. Within our laboratory, we actually have a clean room environment, and then within there we have laminar flow hoods that particles are manipulated and identified underneath of a stereo microscope, and then basically partitioned out to look for different types of analysis. So the preparations that Julia was talking about earlier are all done in a laminar flow environment under a stereo microscopy, using stereo microscopy. and also related to spectral libraries. We have a lot of our own in-house spectral libraries that we have generated. We also use commercially available spectral libraries. Specifically for Raman, we use Know-It-All database by Bio-Rad is our one commercially purchased library that we utilize. And I think for Raman it has maybe 15 or 20,000 Raman spectra. But then we also have years and years of compiling our own internal Raman libraries, as well as the libraries that we've generated on our automated Raman system.
Okay, thanks, Dave. On to the next question. So when dealing with protein particles, how big does the protein agglomerate have to be to recover from the solution?
Typically, the protein agglomerations just need to be visible. So typically, a visible particulate would be classified as something greater than 50 microns. So in that 50 to 100 micron range, typically is something that's easily recoverable and unisolated.
Okay, great. So to our next question. You mentioned Raman spectroscopy is complementary to FTIR. Do you mean to say that Raman is more typically used after FTIR?
So this is Julia. Typically, as an analyst, that's usually the route that I take, is that I'll first perform FTIR on a particle or on a fiber, and depending on the results that I'll get from that and from the SEM, then I'll move on to Raman. But that's not the case all of the time. It kind of depends on the analyst's preference, but I usually look for things, like I mentioned, oxides have a really good Raman spectrum. So if I have something that isn't giving me a very good FTIR spectrum, and then it has high amounts of oxygen and iron in the sample under SEM, then that's when I'd move on to Raman. Or if someone is looking for something like dyes and pigments, that might be a case when I would perform Raman first, because Raman is a very good technique for picking up pigments. So it will depend on your sample a little bit, but in general, I'll perform FTIR first and SEM, and then see what I'm dealing with.
Okay, great. Thank you, Julia. Next question. Someone stated that we have observed several particles in our drug formulation, and we believe that they were from the excipient-drug interaction. We filtered the formulation through polycarbonate membrane and checked the particle shape size with microscopy. We would like to study the composition of these particles, say by FTIR or SEM, but the amount of particles were too few to work with. Do you have any suggestions?
This is Dave. I certainly do. When we're talking about view, I think that it's not clear of the particle size that you're looking at. If we are looking at the visible or subvisible, a lot of times some of the methodologies that can be utilized, such as the automated Raman that I discussed, can be in the subvisible range. Typically that technique takes particles down to about 2 microns. With our scanning electron microscopy methods, we look at things down to about 0.5 micron. So just because something doesn't look during a filtration, doesn't appear to be visible, there are other techniques that can be utilized to interrogate a sample in the subvisible range as well.
Okay, great. Thank you. So the next question, so how do you keep your tools clean to prevent cross-contamination between cases?
This is Julia. So typically when we perform our analysis, each analyst has their own set of tools that they're responsible for. And we use stainless steel tools so that they can be cleaned. We also use low shed wipes for our clean room environment. So what we'll typically do is rinse those tools with DIUF water, which has been filtered to remove any large particulates. And then we will also rinse those tools with methanol and then wipe them down with our low shed wipes. We also use, our scalpel blades have the interchangeable blade holder so that you can change your blade between samples and discard it. But typically that's enough. I'll usually inspect my tools under the stereo microscope also, just to ensure that there isn't anything stuck to them before I start with analysis. But I've never had an issue with cross-contamination by cleaning my tools between.
Okay, great. Thank you, Julia. Next question. The next question is, light obscuration is mentioned in the USP as a method for foreign particulate matter testing. What are the limitations of its application when it is preferred to use that method versus others that were mentioned during the talk?
Well, the big difference is that light obscuration doesn't give any chemical information about your sample. Light obscuration will give you your particle size, distribution of your materials, but a lot of the things that we talked about and focused on in this talk today are foreign particulate matter. So when we're starting our analysis, we do not know what the material is, so we're utilizing techniques that allow us to get down, understand particle size distribution, but also allow us to interrogate a sample and understand its chemistry so that we can identify that particulate. Techniques that are used in the USP for just particle size distribution do not have accompanying capability to do chemically specific or identify what you're dealing with. So that's the significant difference between the two. What we do experience and a lot of our customers do is they will employ light obscuration techniques to understand particle counts of a certain environment. And if the number of particle counts is out of specification for what they're typically used to seeing, then we will interrogate with a chemically specific type of analysis. So they're used as a complementary technique to the techniques that are used to get not only particle sizing but chemical information. So it's very important. It's a very high throughput and a low-cost analysis that gives very good information on particle counting. But when you need to understand what the actual particles are, then you have to move into a different methodology that allows you to get that specific information.
Okay, great. Thank you, Dave. Next question. So what is the advantage of buying a library rather than creating one with in-house particles?
Time and money.
Yeah.
It's important for you to do an in-house library. I think it's important for every laboratory, whether it's a pharmaceutical company, a materials company, or a testing laboratory, to have a good understanding of what their environment is in their laboratory, and to have some indication of the different particulate types that might be present in that environment. When we're looking at foreign particulate, we're often going back and looking at potential source material to identify a potential contaminant against. So always having a library to search your internal particulate against is a very important aspect. Why we utilize commercially databases, the cost of doing 20, 30, 40,000 of samples. For example, I believe the Know-It-All database has over 200,000 compounds in that. So to create your own internal library would be very exhaustive and very expensive. So it's important to have a small library where you can utilize some of the most common things you have in your facility, but the commercially available libraries allow us to expand our scope and look at a large variety of different compounds for a pretty cost-efficient way.
Okay, great. Another person asked, how do you isolate submicron particles for analysis?
So typically anything that we couldn't manually isolate would be filters. So we have filters that can go down to 0.8 microns, and we're just going to pick the filter that is going to have a smaller pore size than those particles. When they're that small, that's when you might move into just leaving those samples on the filters for analysis and either performing, like Dave said, SPE analysis is a good one because those samples are filtered right onto those gold-coated filters that are placed right into the machine. CCSEM is also a good one, or even manual SEM. You can place the filter right into the SEM instrument. And then we could, depending on the sample, you could also just take that filter and put it directly underneath the Raman instrument as well.
Okay, great, thank you. Another question, what brand of low-shed wipes do you use?
Well, I'm not exactly sure, but we have your question, and we'll respond back to you directly with that.
Okay, great. Another question, do you also provide advice on particulate toxicology data and related information?
We don't typically do that, but we do collaborate with other groups, other laboratories that do that. So if that is a particular inquiry that you have, we can certainly point you in the direction of someone that can handle that.
Okay, great. And do you perform spectral interpolation? Interpretation, excuse me, if there is no match in the library?
Definitely. It's done very routinely. When we're dealing with foreign particulate contamination, you typically have an unknown material that's been in your drug product, mixed with multiple excipients, has been applied to different environmental conditions and chemical conditions. So very rarely, when you're looking at foreign particulate, are you dealing with a pure component. Spectral libraries are very valuable when you have a pure component that you're dealing with, but unfortunately, for foreign particulate analysis, that's not usually the case. So it becomes very important that you understand the process of going through something manually. We'll use a database to get a good general idea and classification of the material, but everything then is followed up subsequently with a manual interpretation of the spectrum.
Okay, great, thank you. And just to reiterate, I have a few more questions here that I'm going to go ahead and read, and then we will wrap it up. But I just want to reiterate to everyone that everyone will get a copy of the presentation slides, along with a copy to the replay. So that will be sent to you via email just a little bit after the webinar has ended. I'll make sure that I get that out to everybody. So the next question, someone who's responsible for collecting and characterizing particles found in a process wants to know, I want to create a visual inspection samples for testing inspectors. Do you have experience with visual sample creation?
This is Julia. I've actually done this before. It is very tough to make something like a vial with a liquid and different sized particulates in it yourself. And that's just because you need to do it in a very controlled environment. So it can be done. I would suggest if you are doing something like a vial, you rinse out that vial at least 3 times, shaking it with the liquid in it and then dumping it out and using the DIUF, particle-free water. And then you're going to have to be really careful and do all of your preparations in a clean room environment and under that laminar flow hood to put those particles in the sample. Now that's for something that you could manipulate yourself. It's going to depend on, so my experience is with samples that are about like 10 microns and up. But it can be done. It takes a lot of practice, and my suggestion would be maybe to invest in something like the sized beads that you can get. You can buy pre-sized polystyrene beads or something like that to use in a liquid. It's a little easier than trying to make samples.
Okay, great. Thank you, Julia. Last question it looks like we have here is, from the slide it seems like a specific tool that you may use to pick up the particle. Could you please share the information? Thank you.
Okay. So we use a couple of different tools in order to isolate particles. Generally, forceps or carbon steel scalpel blades are used. For really small particles, you might use a tungsten needle. And if you're only doing an SEM prep, you can actually cut a piece of the carbon tab, lift that with forceps, and use that to manipulate the particle. You're less likely to lose it that way since the tab is sticky.
Okay, great. Well, thank you, everyone. This concludes our Q&A portion of today's presentation, and thank you, everyone, for your time. If you would like to get in touch with any of us, you can contact us at any time via email Also, if you want to get the latest updates, webinar announcements, and more, you can subscribe to our blog, follow us on Twitter @GatewayTweets, or connect with us on LinkedIn. Lastly, if you would be so kind to fill out the quick survey at the end of the webinar, we would greatly appreciate your feedback. And thank you everyone again for your time, and have a great day.